structure of proteins. In this video, you will be able to first recall a bit about the proteins, their building blocks, and then we will learn the different levels of protein structure individually by using examples and illustrations, of course. So, first of all, what are proteins? We all know proteins are made of amino acids which act as a building block for these proteins. These amino acids are connected with each other by peptide bonds. So what happens is, like, when one amino acid comes in contact to second one, the carbonyl group of one amino acid will interact with the imide group of second, and there will be a bridge which is CONH, and we call it as peptide bond. Since it's a condensation reaction, so water molecule will also be formed. This way, the same pattern is repeated regularly, and as long sequences of amino acids are formed, and the protein can have these amino acids ranging from the number from tens to several thousands. Now we will come towards our main part, which is levels of protein structure. Okay, so the first and simplest one is the primary structure, which is simply the long chain of amino acids. It's just a linear sequence of amino acids connected with each other, and this is the simplest form. And as the level gets higher, the complexity of structure and the interactions within that molecule will also get increased. So if this chain of amino acids start getting interactions with the polypeptide backbone, then what will happen? A new structure will be formed, which is a second level secondary structure, in which we will see spiral arrangements and some foldings in structures, just like spiral, like a spiral ribbon, or foldings just like a paper fan have alternate inward and outward foldings, right? Besides of these two combinations, other interactions are also built. Then a three-dimensional structure will be formed, which is known as tertiary structure. These multiple polypeptide chains units combine together, similar to these tertiary ones, and if these each combination of polypeptide chains combine together, then they will form a higher level, which is quaternary one. Now, first of all, we will talk a bit about the primary structure. We all know that it will be one-dimensional, of course. It will be linear. It will be a simple chain of amino acids which arrange and rearrange again and again to form these diverse range of primary structures. Primary proteins are only 20 in numbers. So if we are saying that amino acids will be connected by covalent bond, then does that mean that there will be disulfide bond in them as well? Because technically disulfide bond is also a covalent bond, and if you may already know that disulfide bond is also present in proteins, but no, although disulfide is present in proteins, but it is not included in the primary structure of protein. And when we are saying that amino acids are connected covalently in this primary structure, we are excluding disulfide bond, in fact. So again, we have this chain, this long string of beads, and in it each circle in fact represents the amino acid. It's all these genetic disorders have got some mutations at the primary level of protein structure, so it's very important to know where the problem lies in order to treat it. For example, after studying the primary level of protein structure for sickle cell anemia, we get to know that the defective hemoglobin is somehow the major factor for it. What is the difference between the two? When we are talking about proteins, arrangement holds great importance. So even if they are the same amino acids, the number is same, but since the arrangement is different, these two are different primary structures. Now we will move forward with secondary structure, and do I need to mention that since the level is becoming higher, so interactions will be more and a little bit more complexity as well. So in this, the amino acids start to interact with the peptide backbone. It happens like the local substructures start to arrange along with that polypeptide backbone, forming interactions, and there will be hydrogen bonding which will be built between the amide and carbonyl group of peptide bond. In here, when we are talking that amino acids are interacting with the peptide bond, then we are still excluding the conformations of amino acids. The conformations of amino acids will be included in the higher level, which is will be tertiary one, but not here. We will exclude the the different conformers formed by the rotation of those side chains of amino acids in at the secondary level. There are two kinds of secondary structures. You can recall that the alpha helix and beta plated shears. What are these? These are basically the name for to see earlier. The spiral ribbon one is in fact an alpha helix, and the one which looks like a paper fan, the with the alternate foldings, is beta plated shears. Just and the alpha helix is like more common. It is smaller in size and looks like a rod, while beta plated shears are rare and they are relatively larger in size. And the alpha helix has only a single polypeptide backbone. Intrastrand hydrogen bondings are present in it, which means that the hydrogen bonding will be contained in one single strand, while in beta plated shears the hydrogen bonding is between two or more strands, so it will be inter-strand hydrogen bonding. And since we have said that alpha helix is smaller in size, so it is well justified that the number of resides are lesser in here per turn. So it means that 3.6 resides are in each turn, and each reside is pointed outwards, while in beta plated shears there are more than 3.6 resides per turn, and each reside is in fact pointed inwards. So here is the illustrations of both structures. You can guess the upper one is of alpha helix and the lower one is of beta plated shears. And you can see the interactions, that how these amino acids are forming bonding with the polypeptide backbone. Since these two are repetitively occurring features at regular intervals in a protein structure, so that's why we say that these are periodic structures, because the feature repeat at regular intervals, so that's why we are saying them as periodic structures. Sometimes it happens like these elements start to show folding independently, like they form a unique combination, just like this one here. Alpha helix is sandwiched between two beta plated shears. We call it as domains or supersecondary structures, in which these secondary elements are forming some unique combinations. Now we will move forward to tertiary structure. Okay, so what are we getting new in this level? It will be a 3D arrangement of proteins, and in here the side chains will also be getting involved, as well as there will be prosthetic groups. I mentioned earlier in secondary structure that the conformations of side chains will not be involving in it, and we will study it in higher level, and this is the higher level in which now the side chain conformation will also play a role, and there will be prosthetic groups as well. Prosthetic groups are the group of atoms which are other than the amino acids in it, the non-protein part. So prosthetic groups can also be present in it, which are other than amino acids. Previously we were saying that amino acids are the building blocks of protein, yes, and we are only seeing amino acids in primary and secondary level, but yes, in tertiary level there can be some non-amino acid elements as well, which we call as prosthetic groups. So what kind of basic interactions will be present in tertiary structure to build it up? Right, there will be hydrogen bonding, and that hydrogen bonding can be between the atoms of peptide backbone, and it can be between the atoms in side chains, right? There will be covalent and non-covalent interactions, and also electrostatic interactions between these side chains. We'll know that if a positive and negative charge come together, an electrostatic force will be formed. So there will be hydrogen bonding, there will be covalent and non-covalent interactions, and there will be electrostatic interactions, and as well as the conformations of side chains will also be getting involved in this tertiary structure. And the arrangement of prosthetic group, if any prosthetic group is present in them, then its arrangement, its position is of highly significance, just like these alpha and beta chains. We study this tertiary level of protein structure by XRD and NMR, and you might have learned about protein denaturation, right? That we know that proteins are sensitive to a minor change in the pH and the temperature and other factors, right? So what happens if a minor change in any of these factors happen, the structure of protein get damaged or get changed drastically. Yes, and that change in structure influence its functioning as well. So we all also know that this protein denaturation can be reversible or irreversible, right? When we talk about protein denaturation, it is in fact a damage in the tertiary structure of protein, which can be reversible or irreversible. The first tertiary structure ever determined was of myoglobin, which is the protein to store oxygen, and had one polypeptide chain, 153 amino acids, 8 alpha helical structures, and a prosthetic group. We all know that it have a heme group, which is in fact a prosthetic group. So now moving towards quaternary structures. The arrangement of multiple polypeptide chains build up a subunit, and when these subunits combine with each other, then they make quaternary structure, right? And based on the number of subunits present in that quaternary structure, we can call it dimer, trimer, and tetramer. If there are two subunits of multiple polypeptide chains, then we will call it as a dimer, and if there are three subunits, then we will call it as a trimer, and in the case of four subunits, we call it as a tetramer. Here it is the illustration of quaternary structures. One lighter blue and another one is darker blue, so you see two different subunits in here. So basically it's a dimer, right? And the interactions in it are non-covalent interactions, just like hydrogen bonding, electrostatic forces, and hydrophobic interactions. One of the unique behavior shown by these quaternary structures is that allostatic behavior. Allostatic behavior is like if a small change happens at one part of the protein, just like if a small change is happening over at the top of the protein, it will influence in a way that it will make a drastic change in the structure of the other distant parts of the proteins, just like at the very bottom of the protein. If a damage happens, or if a new molecule gets attached to one side, for example at the top of it, then you will see an instant change at the bottom of the protein at a very distant site as well. This kind of behavior is known as allostatic behavior, and this behavior is because of the non-covalent interactions present in the quaternary structure of protein. And do you know that this allostatic behavior is well exhibited by hemoglobin, right? Hemoglobin, we all know it as a protein which transports oxygen in blood. So do you know that this hemoglobin is an example of quaternary structure? And yes, it contains alpha chains, two beta chains, and four subunits. You can see here four subunits with different multiple colors like pink, purple, blue, and brown. These four subunits show that hemoglobin is a tetramer, right? And it shows the allostatic behavior really well. How? That if oxygen gets bind to one subunit of hemoglobin, it influences a drastic change in other subunits in such a way that subunits also become capable for oxygen binding. So the binding of oxygen to one subunit make it easier for oxygen to bind to the subunits. This is because of the change in the structure after binding of the first oxygen. For the exhibition of this allostatic behavior, the bridge between these four subunits is of great importance. Yes, a bridge which is 2,3-bisphosphoglycerate, which connects these four subunits together, is in fact responsible for this allostatic behavior. And when this bridge is removed, the allostatic behavior will no longer be exhibited by this hemoglobin molecule, and instead the hemoglobin will start to behave like a tertiary protein, which is myoglobin, right? Thank you for watching. If you have any question, you can ask in comment section.